So this is the second video about plant identification, and I'm going to show you how to prepare 5 different plants for pressing in the plant press. So the first thing to remember here is that they have to be a specific size. They have to fit into the size of this newspaper, no matter how big the plant is. This is standard size for plant collection. So I have 2 tools here that I'm going to use. I have a set of pruners, which I'm going to use for cutting woody branches, and then I have a pair of just ordinary scissors, which I'm going to use for finer work. And notice that there's a note on the newspaper here. When you put something in a piece of newspaper, say what it is, where you, the date that you found it, where you found it. Don't just put it in newspaper and think that you'll remember when you come to look at it again after a while. You will not remember. So also you may have field notes, and if so, make sure there's something written on the paper that will link the field notes to the specimen, maybe a number or something like that. So the first specimen here, this is a branch off a tree that's in the backyard. It's a cultivated tree, so obviously I'd write that somewhere. I don't want somebody getting frustrated because they're trying to key it out in the native Oregon trees list. This is an easy specimen. All I'm going to do is take a branch with some leaves and arrange it so that it's the size of the newspaper. I like that it's got interesting branching here. I'm going to try and keep that, so I'll take that off. I'm going to use this as my specimen, this branch here. That's plenty of material, so I can take these others off, but I'm going to leave the base of the stem so that anybody looking at the specimen can tell. I think I'll just take that one off completely, it's a bit in the way. So there we are. I've reduced the size of the specimen without actually losing any information. I have the branching here, I have the leaves, I have the flowers, and I'm just going to flip a few leaves over so that we've got the underside as well as the upper side. And that's my specimen right there. I'll try and separate the leaves and the flowers a bit. So that's what I'll have in the plant press, and when that's glued to a piece of paper you'll be able to see the inflorescences, the leaves and the branch. So that's the first specimen. Now what shall I do next? Okay, here's a weedy grass. It's a brome grass from the backyard. So let's have a look at that. We've got the roots. Unlike the prunus, that was the branch off a tree, so in the notes I'd say it was a small tree. This, we have the whole plant. We have roots, leaves, and this grass inflorescence. And it's way too long. So for this one, what I'm going to do is I'm simply going to fold it like a concertina and do it carefully so that everything is well inside the paper, or anything that matters. So here's a short inflorescence. I'm just going to fold that over. I'm going to fold this over. Still not good enough. I'm going to fold it again. And there I have it. That's my specimen. Make sure that leaf goes inside the newspaper. That's fine for a grass or a sedge, a graminoid. You just concertina them like that, and we have everything there, the roots, the stem and the inflorescence. So that was easy. The third specimen is a weed. It's a sow thistle growing out in the alley. It's not flowering. If it were flowering it would be even bigger, but once again that's too thick, too large. I'll just use the same technique. I'll concertina it, and I don't need anywhere near this many leaves. In fact, I need hardly any leaves at all. So what I'm going to do is snip them off above the base so that whoever's looking at the specimen can see how many leaves there were. I'll leave how many? I could leave 4. I could try doing that. how that's coming along. I've left these small ones at the base. That may be important for identifying the plant. So I've got them coming up here. Let's see how to do this. Let's just keep another one at the top. A young leaf up here. I'm just greatly reducing the amount of material that I have to deal with. And it's leaking latex all over the place. This is a plant that contains a lot of latex. Okay, how am I doing now? I can do the same trick I used with the grass. I can concertina. So I'm going to take this leaf, and I've not only cleverly got it inside the newspaper, I've also got it the other way up, so it's now doing, so I did two things with one action. And then I'm just going to fold that around like that, and that will be my specimen. What I might do is, because the stem is so thick, I might actually just cut it so that it'll maybe dry better. I could use this blade here, for instance, to carefully just open the stem up a bit to help the drying process. So there, you see, it's more open, or I could even just cut it in half. I could just take half the stem off completely. And the same with the root. You know, if the root's a bit too thick, I can just use these pruners and remove half of it carefully, like that, you see. So in this case, I've got rid of a lot of material, but I haven't actually lost any information. You see, the base is still there, the root's still there, but there's only half of it. But the part I removed was the half that wouldn't be visible in the final specimen. So that's 3. It's going to get more and more difficult. We're heading towards fairly extreme botany at this point. So this is the Nandina shrub from the front yard, and this has enormous leaves. So I don't know if you can see it, but, well, anyway, I'll just pull this one off. This is one leaf. So how am I going to convey that information to whoever is looking at the specimen? They may be able to call me up and ask me if they really want to know, but it's possible I will have been dead for 200 years already. So let's see how I can go about doing this. Well, I'm going to take a branch. Let's start here. So it's I'm going to have, I like this leaf. This is a well-shaped leaf, not too damaged, so I think I'll just keep this one. It's a little bit smaller than that one, but I sort of like this branching pattern. It's unique to the shrub, so I'll hang on to the branch with the leaf bases, and now I'm going to try and get this to fit the newspaper. So I'm going to reduce this by basically taking away half the leaf. So I'm not going to just cut it right down the middle, I'm going to remove it in such a way that you can see where the different branches of the leaf were before I cut them. So you can see that there was a branch here that was just the person looking at it will assume that it looked like this one, and so on up the leaf. So will it fit yet? Let's have a look. That's kind of awkward, but we can twist it around a bit so it's more or less flat. So let's see how we're doing. We've got that. So I'm going to reduce the size of the inflorescence a bit. It's got a branch there. I'm just going to take that off, leaving the stalk that goes to it. And what else shall I do? That's pretty much, I want this inside the newspaper. Can I do that? I think so. So I'm going to remove this as well, trim this leaf down a little bit, and then use this to be the top side. Oops, it broke off. Oh well. I'm just going to put that in there as the top side of the leaf. It's a little bit messy, but, well, you know, it's a difficult specimen. This, I'll just fold it back so that it's inside the newspaper. So something like that, maybe. Okay, so, once again, this is a bit thick, so I might cleverly try and cut it down the middle in such a way that it's still there. You see it's now much thinner, but it's got the same information. The branch is still there on one side. So I'll probably go with that as my specimen. It's not pretty, but it's got all the information I need, and fold that over a bit so it doesn't stick out of the plant press. So that's the way I've just reduced the size of a very large leaf. Finally, the fifth specimen is this plant called Jack in the Pulpit, which is invading my backyard. So how am I going to deal with this one? It's slightly too big for the newspaper. The root is a bit big and fat, so I need to reduce the size of the root. I'm going to try and cut it down the middle. And the root here is very important because this is a plant that's potentially invasive, and if you look, I don't know if you can see this, but if you look at the root, you can see that it has these little balls. These are, it's actually a stem, and this is a branch that can fall off and form a new plant, and that's why it is so invasive. These things can get all over the place and they'll be in the soil. So I want to maintain that piece of information. Now I want to keep the inflorescence, obviously, that's the key part, and I'll need at least one leaf. I'll keep this leaf, and once again I don't have that much space, so I'm going to cut it, remove some of the tissue. I'm going to simply cut the blade off, not right along the midrib, but just next to it, so that the fact that there was another side to the blade is clearly visible. So I've just got one leaf here. If I want both surfaces from this one example, I'm going to have to flip it over. I could do that, or else I could just put this on the a piece of this on the newspaper with the lower side up. Yeah, that leaf, there's not enough room for it. I'm just going to take it off, leaving the leaf stalk. Right, now I have this amazing inflorescence. It's got a huge specialized bract called a spathe, and then an inflorescence inside of it that is highly specialized. It has unisexual flowers. It's got a male part and maybe a sterile part and a female part. Once that's pressed, it's going to be hard for anyone to see the inflorescence. So what I'm going to do is I'm going to carefully cut it open. Here we go, to display the flowers. So right, I've cut it. So look at that, you see, look at all that information about the inflorescence that would have been lost if I just pressed it flat. Look at this thing. So it's got female flowers, male flowers, flowers that are modified into hairs. These have a very specific function, and then a sterile portion. So this attracts insects looking for dead meat, I believe, and they're going to crawl in here, get stuck, by the hairs, which are filling the throat of the tube, and then they'll walk around in here and pollinate the flowers, and then finally this will fall open and they'll be able to escape and pollinate something else. So that was so I was able to open this up, and I still have the shape of the spathe, but I've also exposed the flowers. So we've got the root, we've got these very unusual flowers, and we have a leaf. If we can get it to behave itself and fit in the newspaper, I'll put it over here. So all of that, and I'm just going to use one of these pieces I cut off to be the underside of the leaf. So that is, there's a lot of information in that specimen. This is the weird structure for the inflorescence, both sides of the